Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. We are very excited to bring you a new episode of the podcast. Before we dive into this one, we have a few announcements to make. The program of the upcoming third edition of the Dr. GPCR Summit is taking shape. This year, the meeting will be held between October 10th and 16th. The meeting will be virtual so that we can allow as many participants from around the world as possible. We will host live talks, which will be hosted on Zoom from October 10th to the 15th. Everyone is welcome to participate by either listening into the talks, presenting a poster, or submitting a pre-recorded talk. We will also have prizes for trainees for their great presentations. We are also trying out a few new things. We'll host networking and poster sessions for the first time on Wonder. We will have also a full day dedicated to trainees and to trainee talks. We'll be hosting also workshops on data analysis, on how to preserve data integrity in the lab, and how to use GPCR DB. Everyone is welcome to attend. And it's free when you become a Dr. GPCR Ecosystem site member, which is also free. Speaking of the ecosystem, we are excited to share that the Dr. GPCR Ecosystem 2.0 platform is now open. Visit drgpcr.com to explore the ecosystem. The ecosystem is your GPCR-focused virtual playground. Join over 50 of your peers who have already registered as site members. You'll also have the option to select a plan once you're a site member. You'll get access to all things Dr. GPCR, including access to new podcast episodes before they even get released to the general public. You'll get access to our new group discussion and forums, our job board, and our learning center, while you'll be able to take a course or even prepare and share a course of your own with your colleagues. You'll also be able to discover GPCR companies and much more. Take advantage of everything that the new GPCR dedicated online playground has to offer today. Once you're a site member, you can also become an ambassador, which means that you'll get your own dedicated Dr. GPCR Ecosystem affiliate link. And every time someone subscribes for the year to the ecosystem, you'll get compensated. This will potentially help you offset the cost of the yearly membership. Last but not least, we're also looking for content creators. Subscribe to the ecosystem and start writing your own GPCR-focused content, share it, and show off your GPCR talents. Visit drgpcr.com to find out more about all our activities. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I'm delighted to have with me Dr. Christel Menet. She is the CSO of Confo Therapeutics. Hi, Christel.
Hello. Hello, Yamina.
It's so nice to have you on board, and I'm glad that after all the rescheduling things that we've done, we finally managed to sit down.
Yeah, it's nice. I'm happy to meet you for the first time. We have chatted during conference, but it's the first time we see each other, so it's nice.
Yes. And it's funny because I follow you on LinkedIn, and my guess is it goes both ways, but we never got a chance to.
Yeah, I was pleased. What was it? A couple of years ago when I discovered Dr. GPCR, it was a nice, finding to see a bit of the community and what's happening in the community of the GPCR itself. So I think as a company really focusing on this type of target is nice to have all the experts, together and see what's happening in the world, and having this newsletter with different conference. For the group, it's really, really useful. I have to say that to know, where to find the conference, because it's not always easy to find the right links.
Yes, agreed. And we're taking the concept to the next stage with the ecosystem where you can actually sign up and become a member, and we're really curating more information. So you would have a page where you can look up the different conferences. It's listed out, you click on it, and it takes you to the link to the registration page, for example, of the upcoming conferences. Which I think is really important to have, because, for example, for the Dr. GPCR Summit, we want to make sure that it doesn't overlap with other conferences, and that's completely selfish for us to know. But in the meantime, since you're gathering the information, why not put it out there?
No, you're right. Yeah, it's a good point.
So let's start at the beginning. Would you please walk us through a brief overview of your career, from where you started as a scientist to where you are today?
Okay. So I'm a chemist by training. I'm actually not a biologist or a biochemist. So I'm a pure organic chemist. So I started my career in Manchester in the UK as an organic chemist. I did a PhD. I was, tried to optimize some chemistry reaction. So I was really far away from all the medicinal chemistry or understanding even, the use of the drug towards therapy. And then I moved to Evotech, just a CRO. After my PhD, I decided not to do a postdoc for a strange reason. But, when you're in the UK, being French, I felt actually old doing my PhD because in the UK at the time, they had only to do 3 years and then they could start their PhD. So they were all really young. And myself, I had to do my 5 years and then do my PhD. So I felt really old and I thought maybe I don't have time to do a postdoc. So I will go straight to company. And at the time, more than 20 years ago, there was a big demand for chemists because it was the time where a pharma company were thinking that, we need to do this huge library to find hits. They did not really understand, chemical space or too much at this stage. I really thought, if you make millions of compounds, you will find hits. I think it stopped after a couple of years when they realized that was not the way to go. So I was recruited by Evotech at the time. Stayed a couple of years and I realized actually going to Evotech really as an organic chemist that behind these chemical reactions, there were drugs. And I started being really attracted at what was behind this chemistry and these drugs and how they start working and how they would activate target or block targets. And then I decided it was time for me to move to this type of environment. And I moved to my first company, which was biotech, which was called First Pharmaceutical at the time, which is called Domain Therapeutics today. So It was my first contact with GPCR. And if I have to say, at the time, it was in 2004, 2005, there was only one structure of GPCR. So it was really early time on GPCR. We obviously knew there was activation, change of conformation, different signaling, but it was such a low understanding of GPCR. Now, if I look at where we are with all the structure of this, these people generated and all this knowledge that were generated over the 15 years, it's amazing actually what was done on the GPCR. It was a really interesting start. I didn't stay too long for really personal reason. My partner moved to Belgium and I had to find a job in Belgium. And then there was a company called Galapagos, a biotech company, which was created and looking for chemists at the time. So it was a good opportunity for me to move there to that biotech. And I really made most of my career at Galapagos. So I moved really from senior scientist to head of medicinal chemistry. I'd have to say that I've seen a huge number of projects at Galapagos, but my main program were on kinase, not on GPCR. But if I compare today, where I'm really back to GPCR, and if I compare kinase and GPCR, kinases is really around the chemistry in the way that you have a scaffold, you can play Lego with it. So you have structure, you have a scaffold, you go and you superpose your structure, you superpose your scaffold, and you can, as a chemist, you can play Lego and find new binders, new inhibitors. Yep. You wish GPCR was the same. Not at all. You have 3 structures of the same GPCR, active state. You try to, superpose them. Doesn't work. Just GPCR have their own lives, their own binding, their own unders, pockets that they form with each ligand. You see that the water are playing such a big role in GPCR to really give this different conformation that is not possible. So now you're not looking at the chemistry, but you're really looking at the receptor. So you're not looking at the enzyme like for the kinase, where you can look at just the chemistry. You don't need the enzyme in the end. You just focus on the pocket. For the GPCR, you need to look at the GPCR and then later on the chemistry, what can fit in that GPCR. So it's a really different way of looking into this type of target. So yeah, this is summarizing my career. So when I was at Galapagos heading the medicinal chemistry, some headhunter came to me and said, look, there is this company that is getting created with that exciting technology, which is coming from Stanford with Brian Kubica, Nobel Prize, and Ian Steyer that have these nanobodies and brainstorming together, they come with these nanobodies and that they call confobodies to stabilize this GPCR in different conformation, and they're looking for the chief scientific officer. And do you want the job? And I was like, whoa, exciting. You go with a white page, you have, there were 6 people in the company. So you go with, to create the team, to create all the drug discovery. And I say, yeah, okay, let's jump, let's go. And today I have to say that indeed, it's a great technology. We are 65, I think, today. We have our first compound in phase 1. Which is really exciting time when when you start testing volunteers and when you start seeing the first data coming out. It's really so exciting. So it's I think summarize where I am today.
That's so interesting that you started out as a chemist and then you kind of got drawn into the GPCR field, and which is kind of amazing. Well, there is a challenge in the GPCR field because these receptors, these proteins are so difficult yet so amazing at the same time, which I think it's what all of us somehow are attracted to, is the level of difficulty and complexity and the beauty of it.
Yeah, the beauty of it, and also you actually have reward on the complexity, because when you find something, it actually works. If you look at the success of GPCR today on the market and the impact they have on patients' life, you think, okay, I'm doing science that is going to work. It's going to help these people, because this GPCR indeed say each GPCR is on his own life, his own way of working. But when you understand a bit of it, you actually really can make a change. And it's what's exciting about a GPCR also.
Yeah. It's funny because you mentioned, that it has its own life. And every time I worked on a new receptor, I always told people, and I think people were looking at me in a bizarre way, I have to get to know the receptor. I really get to know them, or him or her, whatever, whatever pronoun you want to use, because you need to figure out in what's the context in which you can start screening or looking at molecules. And you form this personal relationship with this receptor because the residues, if there's a structure, you can kind of see it changing depending on what the mutation is. And you develop that personal relationship with your target.
Yeah, that's true, actually. And, indeed, you need to know in which host you want to be expressed, what signaling you want to be read out.
Exactly.
So it's indeed, it got his own life or his or her, but it's which is not the case at all with many other targets. I will not, I worked on many on kinase. I worked also with ion channel, but as a chemist, ion channel hasn't been a success, let's say. but yeah.
So let me take you a step back a little bit before you went into chemistry. As a child or as a teenager, were you always interested in chemistry or in science in general, or what did you want to become?
I wanted to become a doctor, but a medical doctor. Mm-hmm. And then I realized that I'm a bit scared of needles, so it doesn't work. But I always like science. I have to say that maybe when I was a teenager, I was more attracted by math and physics. But then when I went towards university, in France, you can start your university and you can have more general view on if you go to, you can go to university or you can go to what we call classe préparatoire, which is to go to the kind of high school that we have in France. Yeah. And then in this classe préparatoire, in fact, you have the choice between physics, math, and chemistry. And so you don't have to choose from the start. And I realized that the chemistry, maybe because I have more a manual side also, I really like, in fact, and I realize more and more that I really like the lab work. And as a CSO, it's what I miss the most. Because even when I was head of chemistry at my previous company, I was still going to the lab. And people would say, but you're mad, you have other things to do. But I really, really enjoy the lab. First, it's the best way to interact with a scientist. You're in the lab, you have a lot of fun and you can discuss a lot of science. And also, I always needed this maybe concrete way of creating things. And the chemistry was giving me this concrete experience of creating things, which math and physics were not. And I think it's what attracted me the most in chemistry. And it's why I moved towards the chemistry part. Mm-hmm. But at the time, I have to say that I was not yet realizing, the impact I could also have on people's life. And it was not maybe so much what I was looking for. I think it's really when I start moving to biotechs and to companies that I realize, but this chemistry, I need to make something out of it. I need to make it, use of it, because, and it's where I understood that you can also impact patient life and really do something good about your chemistry.
Yeah, and it's funny you mentioned chemistry, and it's a long story. I was talking to someone yesterday, and we were looking in VR at the structures of GPCRs and stuff. And I remember this quote from Breaking Bad when Mr. White says that the chemistry must be respected, and I thought, and it could be translated to the GPCR must be respected. When you say you have to get to know your GPCR. But, you raised an interesting point about, understanding that the chemistry can have an impact on people's lives. And for me, that happened during my postdoc, thinking actually biochemistry and GPCRs and everything, it's just quote unquote chemistry. But how beautiful that contact is, that you go from the chemistry to the protein to the organism and then to the human. It's just, I think it's marvelous.
Yeah, and you never get tired of it. When you tell people, you try to communicate what you do every day, or that people cannot realize when you say you work in pharma companies, they say, oh, pharma is not, it's not a good business. First, they don't realize that scientists, they just do it by passion. They're passion. They don't do it because, they're going to a lot of money. So the scientists are not the one making the money. So it's not that. The natures, how he works in the body, you never get tired of it. It's so, so much to learn. So every time I discuss with my scientist, I find it, yes, the passion of science is always around.
Yeah. And we also have this crazy resilience, because 90% of the time experiments fail. But then you go from dopamine rush to dopamine rush. But the question is, what's the distance between the two dopamine rushes when something finally works?
Yeah, you're right. It's gosh, yeah. If you think about all your career, what works and what did not work. Yeah.
Yes.
But you forget what doesn't work. Huh? You always forget.
Yes.
You get so excited by what works that you forget that it did not work for the past months.
Exactly. And then you also learn what doesn't work, which I think is also a very important thing to do in science. You need to figure out what doesn't work with your receptor in the assay, or what doesn't work in the chemistry, in order to move past that obstacle.
Yeah, indeed. And if we were a bit, as a scientist, a bit more also focused on what doesn't work, maybe some publication on what doesn't work would help us to learn more also, because it's true that if we look at what's published is only on what works, but sometimes, you end up repeating things that people have tried before, and you will just end up with the same conclusion that it doesn't work. So it would be so nice, in the end to say, okay, there is this publication, and guys, is to tell you that this we tried, there was a logic behind, there was an hypothesis, but it just doesn't work because, and we believe because of this reason. That would be actually great also sometimes.
Yeah. It's two different thoughts came into my head at the same time. As a PhD student, a lot of things did not work. And I had a friend who happens to be French, and we were all joking, okay, so we should start the Journal of Negative Results, which I think could be really awesome to have. But now that you're mentioning it, I think maybe that should be Dr. GPCR's next project, is to, because I was every year I think about a new program. What else can we do to serve the GPCR community? So we started with the podcast, and there was the newsletter, and then there was the summit, and then the virtual cafe, which is a once a month scientific webinar. But I think, becoming kind of the +1 GPCR, the Journal of Negative Results, where people can actually put it out there.
Yeah, it would be really good, because sometimes, also some well-known GPCR people claim, my compound is biased, and it's because it's activating this signaling. And you try to repeat this data, and you cannot make it repeat. And then you go to a conference, and someone is claiming that again, and you start chatting with the people around, especially now they were, online. And then people tell you, hey, it's not working for me also. Yeah, it's not working for me. And then you realize that many people have tried, and it was not working. So there is something, fishy about this data. And it'd be nice that, people start, bit saying, I tried that, but it's just not working. And if someone confirms, we tried also, and it's not working.
Yeah. Yeah.
And maybe the one that made it work can explain why you worked in this case, and that will be also a nice way forward. So
yeah, you raise another interesting point around, being able to have those discussions. I think the ecosystem on the Dr. GPCR side, and this, the podcast wasn't meant to be advertising the ecosystem, but I think it's an answer to so many questions. Being able to have a forum about a publication, about a result, about a receptor is important. But on the other hand, I think when you publish papers and you look at the materials and methods, there is sometimes a limited space. So there is a detail that most of the time doesn't end up in there. One, not because people don't want to put it in there, it's just because they don't think about it. You make a movement, you do tissue culture, the way you deposit the ligand in the well, something makes it so that you get that data, but you don't get to share it, or you don't have the opportunity to discuss that detail. And maybe that's the missing detail, or maybe the buffer component, magnesium, calcium, whatever, changes everything. And being able to share that with the world is really important.
Yeah, I agree. Yeah, indeed. And maybe, we gain more time, and we can help more people, because
yes.
It's a bit a shame that we keep repeating data that's been done before and will end up not working. So yeah, indeed.
Exactly. So let's take a step back and go into your first contact with GPCRs. You mentioned that it was at now what known as Domain Therapeutics. What drew you from Evotech to the company?
It's really what drew me to the first pharmaceutical at the time. It was really, I wanted to go in medicinal chemistry. I wanted to really understand why I was doing this chemistry. And I was not feeling like going to a big company. I did not, obviously, I did not have experience of big company, but I was just hearing of what people were saying, that, when you were going to a big company, but you were a chemist, and then you will stay a chemist. And me, I was seeing myself, if I'm a chemist somewhere, I want to see what's behind the chemistry. I want to, be able to contact the biologist to understand what they do with my molecule. So when I discuss with people around me, they say, but you need to move to a small company, to biotech. And then there was indeed, at that time, it was not GPCR as such. It was really Force Pharmaceutical was looking for someone, were looking for a chemist. And I just sent my CV, and things went really well. And I discovered then the GPCR. So I went to the GPCR. They did not attract me, but then I discovered them. And yeah, it was really, I remember the start, and we were really looking a lot of that rhodopsin structure, try to, doing homology model, and we were working on class C at the time. So I can tell you it was far-fetched, and I really understand that, yeah, this is a really interesting type of target. How can we, especially the class C, which I haven't been going back to yet, because they're a bit of a special type of GPCR, but yeah, it's really, it was a start, and with limited tools for GPCR.
Yeah.
And then after, when I moved to Galapagos, I worked on a number of GPCR, because we were, target agnostic. It was more type of therapeutic areas that we were focusing on. I was not so successful, I have to say. So at Galapagos, we did not have a specific technology for screening or to approach this target. And most of the screening I done, the one I worked on, I would say that Galapagos still had a couple of GPCRs that were moved to clinical stage, but the one I worked on, I was not successful to find any hits. So when I was contacted by these people that say, okay, there is this company, that great technology that will help to find hits, I was like, oh, interesting, because I was not successful to find hits. We screened almost millions of compounds and nothing came out of this screen. So what's about your technology that you want, to bring on GPCR that will help to find this hit? So I think it's what attracted me also to Confo, the fact there was a technology to find the small molecule that I was not able to find in that company I was in.
How I'm gonna try and formulate this properly. Like, what's the percent of interest in GPCRs that drew you to stay, quote unquote, in the field and continue working on GPCRs? Or was it more for you from a chemistry standpoint, being able to make molecules that you can screen and get hits in order to have these molecules end up in people? Like, is there a separation that you can make and say, well, I don't care about the target as long as it's interesting and I can have hits that will potentially end up in the clinic, or it needed to be GPCRs because you were not successful at Galapagos with that screen?
I wouldn't say that it would be any target. I have to say that if I look at my job today, it's not the chemistry I enjoy the most anymore.
Okay.
It's really the GPCR I enjoy the most, understanding how they function, the biochemistry of the GPCR, understanding, once we have the molecule, the structure, looking into the pocket, understanding the movement of the TM and looking at the signaling and try to understand, okay, why, we're activating this signaling now and why we're seeing this effect in biology. So if I look at my job today, I would say the chemistry, the molecules themselves, maybe it's 10% of my interest against 90% for the GPCR itself. Because I mean, going with this target, I realize, I really enjoy any type of conference I go to the GPCR now. If people don't even, talk about molecules, I don't mind. And especially that now we have also biologics in the pipeline. So sometimes there is no small molecules involved. So even if I'm a chemist, I still think like, gosh, it's so interesting what you're just talking about. Or, so it's, no, it doesn't have to be chemistry anymore. I think the GPCR really took my mind away from the chemistry.
So in short, you're hooked.
Yeah, exactly. I was hooked by the GPCR.
There you go. That's amazing. Do you have a favorite one that you feel like?
Do I have a favorite one? It's a tough one.
It's interesting.
I have a class. I really like the class A so far, but maybe because we have the main focus, we have few class B, but we have main focus on class A. And so far, I have to say that the class C, I haven't been back to that interest yet. But a GPCR itself, difficult.
It's a tough one, right? Yeah. It's difficult. The reason, so I always ask this from my guests, and it's interesting to see what people answer. So some people will say, well, I don't have a favorite one. I think of them as tools to better understand GPCR biology in general. Others will say, well, GPCR XYZ, or just one. Others will say, can I pick a G protein or a beta-arrestin or something like that? And earlier this week, we took it to the next level with Andrew Tobin when he asked me, he said, well, I do have a favorite GPCR, but I also do have a favorite phosphorylation site on my GPCR, which I thought was, taking it into a new dimension.
So I will take it differently. I have our most advanced compound is on the angiotensin type 2 receptor. My question is that, is it a GPCR? Because it doesn't bind to G protein. So maybe my preferred GPCR is actually not a GPCR. It's maybe a 7TM. Exactly.
And it's funny because to that point, somebody told me, well, I do have one, but it doesn't couple. We were talking about TXR7, a chemokine receptor that doesn't couple to G proteins. And I said, well, I ask this in the context of GPCRs because it's the Dr. GPCR podcast. But it would have, and when I came up with the name of Dr. GPCR, I thought, okay, it doesn't have to be a GPCR, because sometimes people take it literally. It has to be a GPCR. It cannot be a G protein or something like that. But from an outer perspective, it would have sounded worse to call this the Dr. 7TMR podcast.
Interesting.
So
there was a company called 7TM, no? I don't know if it still exists, but there was a company called it.
I think so. I think they make some antibodies or something.
Oh, they still exist? I don't know, but I know there was something with 7TM.
Yes. So, but it's all of this to say that there is no right or wrong answer. All the answers are acceptable, and whether it's a receptor that couples to a G protein or doesn't, as long as it has 7 transmembrane domains and does similar things to the rest of the GPCR family, it's an acceptable.
No, but if I'm honest, actually, I think it will be beta-2-adrenergic receptor. It's a simple one is the one that's been well studied, but if I think about it, is the one that for which I learned the most, because it was, when I joined Confo, it was really the beta-2-adrenergic receptor that Kobilka and Jan, push forward. They solve all this impressive structure. these conformobodies and also with a G protein complex. So, and I learned so much by looking at this structure to understand how the conformobody was binding. And I must have spent the most time on that receptor, even if it's not something, we try to drug or anything. But I spent myself a lot to really understand the biochemistry of the GPCR and really understand also the technology of Confo. So maybe, if I'm honest, it would be a beta-adrenergic receptor.
Yeah, that's I think that's a good choice, and that's an interesting choice that sometimes people, dismiss because, quote unquote, we know everything about the beta-adrenergic or the beta 2.
I don't think we know everything, but we know some of it.
Exactly. I don't think we know everything. That is correct. But it's a great model receptor.
It's a great one, and it did allow to learn so much, on GPCR and the different conformation. I think it was really a start of a new era for the GPCR, these new structures that on with a beta-2 adrenergic receptor.
Yeah,
they really changed the full view on GPCR that we have today.
Exactly. When I think about structures, I think, about Palchevsky's bovine rhodopsin in 2000, and then the next one that comes into mind that really gave out a lot of information is the beta 2.
And if you think about all the companies were created after that.
Exactly. And all the structures and, let's just, if we want to take it back and make a human type of thing around the beta 2, we know him or her really well.
Yeah, indeed. Yeah. It's almost a friend. It's a family friend.
Exactly. So we you mentioned, not doing a postdoc because you felt like, you were too far along or too old for that. Didn't want to use the term, but let's just.
I was still young, so you can use the term. Now you cannot, but at the time.
I always tell everyone on the podcast and even outside of the podcast, I've been 25 plus for some time now.
Yeah, that's true.
Yeah,
you're right.
So it's fine.
I'm still 25 plus, and I should be here for a while, hopefully.
Yeah, me too. Yeah. I'm 25 plus for a while. Do you regret the decision of not doing a postdoc?
No, I don't actually. I really enjoy my PhD, and I really enjoy my experience in academia. But I have to say that I also really enjoy my experience in industry. I never felt, some people feel, in industry, you need to do what your boss wants you to do and what the company strategy wants you. But I would agree and I would disagree also, because you can run the science. Obviously, there is a strategy for the company. There is an aim for the company.
Of course.
But you can bring that strategy and you can bring to that same aim the way you want and bring the science, you want to drive, you can drive your science the way you want. And it's what I try to give as a spirit and culture at Confo, that, yes, we all scientists come up with ideas. You know where we want to go. So come up with your ideas. Let's develop the science all together to bring us where we want to be. So it doesn't have to be that, you follow what I say or you follow, what's described. Let's think out of the box and bring it where we want to be. So I don't think a company prevents you to be a scientist. There is indeed a common goal, and that we need to keep in mind, because, it's the way it is. But sometimes it's also maybe the fact that we have a common goal, the science goes somewhere, because sometimes in academia, people can get lost also in the focus and trying too many things and get lost in what they're trying and forget, what they wanted to demonstrate at the start. So, sometimes having a goal can help also to better drive your science also.
Agree. And you made a bunch of great points. And I think it's important, especially for those listening who are still in academia, potentially trainees who might think that, you don't want to go to industry because it's the dark side.
Yeah, indeed.
And I think it's this bell-shaped curve type of distribution depending on where you are. Big companies, it's different than smaller biotech companies. And I think over the years, the landscape changed a lot. So I think you're right. You need a goal no matter where you are. I think you also need a personal goal in your life in order to give you a direction.
Yeah,
and unfortunately, you don't learn that at school. You have to kind of figure it out for your own. But in a company perspective, I really liked that you mentioned, that you're still a scientist, science and the data drives innovation. And yes, there is a goal, but there is, I think, a safe circle around that goal where you can go explore, and who knows, maybe you can put some of your time towards an interesting project that is high risk, but then if you generate the data and you make a case for it, you might even change the history of the company.
Yeah, indeed, completely agree. I mean, if I see where we started and where we are today, and the evolution of the technology of Confo, and we were only focusing on small molecule. Now we can find agonist VHH or antibodies.
Mm-hmm.
So we really completely changed the technology, and we had to do all this proof of concept and really adapt the technology to be able to generate all this. So and this is coming from the scientists from Confo, coming out of the ideas and the great mind of been doing a PhD, sometime a postdoc, and being able to think for themselves. So it's why I say academia, my PhD, I really enjoyed because it made my brain. But arriving to company, I never felt like I would not be able to use it anymore. It's the opposite. Maybe it's because I was in the biotech world, I don't know. But what I know also that I had the money to do what I wanted to do. So that's also make a difference. In a company, you have a drug, you can test it, you will test it in vivo, you will test it for the properties. In academia, all this costs a lot of money, and for a company also, obviously, but we get the money to do it. So I think it's a main difference. Sometimes, when I'm part of a lot of review of grants for some from some government things in Belgium, and sometimes I receive this grant where, in academia, they want to do all this drug development, which is hugely expensive. And I was just thinking, why not, collaborate with a company to do so and do more, basic and exciting science in a university, because I do believe that universities are really good to find new things, to find exciting, and then the drug discovery should be kept, in the company. Not in the research, but really R&D type of approach where it's, you pay for it, you have the money for it, and it's all, a well-known process in a company. So, but yeah.
Yeah, I think it's, and it's interesting the way you put it, and it's kind of my view as well as to science in general. I think universities are really good at nurturing basic science, making discoveries, better understanding processes. And for a long time, there was this distance between private sector and academia. And I'm glad to see it like that seeing that distance shrink, because I think one enables the other.
Yeah,
and it's really a distribution of talent.
Yeah, you're right.
And you need different tools, different approaches in order to go from discovery at a basic level, basic mechanisms in academia, to a drug that goes into humans.
Yeah, you're right.
And it's really
different mind also. I think when you have this discussion with this word experts working in academia, I really enjoy it because they have a completely different view of I really, for me, we should, as a company, never get away from university. I really enjoy all the collaboration we have with academia. It's really nice collaboration, I have to say. And the people also in the company really enjoy this type of collaboration.
Yeah, and it's beneficial for everyone. And I think the main goal out there should be improving human health.
Yeah, indeed. Yeah, the common goal. Yeah.
Exactly. But we all have our roles to play in that big picture.
Yeah, you're completely right. Yeah. And I think we're coming towards that, because you see things evolving. I think in the past, it was not so much collaboration between. I mean, when I did my PhD, I was not collaborating with companies. It was really, obviously, it was chemistry, maybe pure chemistry. But I see it now. Like we have one of our employees that wanted to do a PhD, so he's doing a PhD at Confo, and we have a PhD with Jan Steyert, which was one of our founder. So, and we now want to submit a grant to get a new PhD student that will be also a part of one of the projects of Confo. It's something that we really try to build around us, to have this student that can learn from industry, but can also learn from the academic side.
Yeah, and I think you make again another great point as to these are merging, and it wasn't the case when I was doing my master's and my PhD. This was the concept was the dark side is the private sector. And that's that. But I think it's merging, and I think it's important to be able to, get work experience in a private company as a trainee.
Yeah, indeed.
Because you learn the science, but you also learn how to set goals and think about your project in terms of goals and deliverables.
And team working also.
Yes, because
which is key.
Yes, but let's face it, that in academia, teamwork is very vaguely defined.
Yeah, you have your own project, and sometimes it's what I saw people coming to Confo, that they had their own project all their life, and coming to Confo and working in a team. And I'm really a team. I like teams. I really enjoy discussing with people, when listen to their ideas and sharing ideas, and and this is the part I do prefer, I have to say. So I have really difficulties to see people that want their own project and one with their own project and they want to share. I'm just like, come on.
Exactly. But it's unfortunately what academia for a long time has nurtured. And I don't know, you've probably heard the horror stories where you ended up in the same lab and two people were put on the same project, and que le meilleur gagne. Whoever gets there first publishes, and the other one, too bad. But I think that's changing a lot.
Yeah, hopefully it does. Yeah. And I think the new generation is understand the need, actually.
Yeah. Yes, and I think so. For in academia, or at least for training purposes, there are steps that you need to go through. You have to fail. You have to make your agarose gel with water at least once, and figure it out when you're sampling.
If you say so, I don't know. I never made one.
That. Never ever had to. I did it once. I don't know how, for the 10 millionth time, and I did it with water, and I was wondering why my samples were diffusing in the gel. But you have to, I think you have to make those mistakes, and that's part of your training. But at the end of the day, if you want to understand a biological event, you want to understand how your GPCR is working, the goal is not for you to prove that you can pipette. The goal is to work with other people, because you can't do it all. And the final result is pooling together all that data from all those perspectives in order to answer a larger question. And that, for a long time, at least in my environment, or in my view, and in what I was seeing in academia, that was not the case. Because then egos come into play, and then the question is, who's the first author? And that's the whole thing. But I think in general, things are improving, and having more collaborations with biotech and pharma and all of these players together, it just opens up to working on higher level goals.
Which is good, yeah, indeed.
Yes. Yeah. So do you think GPCRs are still good drug targets? I know the answer is typically yes, but if you could expand on that. Yeah,
I think they're still really great. When you see first still the every year outcome of GPCR, almost every year you have a new drug of a GPCR, so they're still good target. And look at the drug today on the market; they are efficient drug target. So they have an effect on patient life. And, I think once in your life, you would have to take a drug that is activating or blocking a GPCR. Everybody in his life would have done it at some point. So there is no doubt. And the GPCR are still so vast. There is still some GPCR. We have no idea what they do, how to drug them, how to approach them. So there is this, dark family that people are not yet coming close. But at some point we will, because we will find the tools. I'm sure there is future great scientists that will have come up with the idea of how to tackle this GPCR that have been in the dark for a while, and nobody's been knowing how to approach them. That will come. I mean, when I see how science is evolving, that will come soon, because there will be great minds that will be able to do it. So I don't think we're at the end of the target. Maybe it's a start, actually. If you think of it, maybe it's a start. Because everything started at 2007, when the next GPCR structure came out. I think it's when it's really started for GPCR. I think before that, it was luck, because, we knew the ligands that was targeting the GPCR. We were making a drug of it, or we were finding a drug having an effect, and then we realized it was targeting a GPCR. But since the 2010 and 2007, we are making drug. We are designing drugs that are targeting this receptor. And this is still really early. It's been like 10, 15 years. So
exactly.
I think we have a good number of years to go still for.
I think so too. You mentioned, tools that we might need to develop to get to the point where we can explore other targets. With the toolkit that we have right now, what else, if you had a magic wand and you could create a new tool that would help, study Class C GPCRs or something that you're really passionate about? What does that look like for you?
It will be a tool that, allow really great expression every time.
Mm-hmm.
Because if you think about the dark side of GPCR, sometimes you cannot target them or understand them because of low expression of a GPCR. And you realize that a lot of data and literature are generated by a low expression and can be completely wrong because the host cells they used at, other things on it that, misled the scientist. And I do believe if we had tools that would help GPCR to highly express every time without, have to change the structure or doing mutation, but really a tool that allow this expression, it will really help already to understand, okay, what this GPCR is doing and how can I tackle it? Because that's the main thing with this, interesting targets. Quite often they're low expressed, but they have a huge impact on your life. But that it's if you can find a way. And also we could work on GPCR in the endogenous system, that will be perfect. So that's, because GPCR, it's nice to have this HEC, CHOS, and all these type of cells. But in the end, GPCR has a way of working in his own cell also. There is a GA protein expression, there is, lipids on the membrane, there is the environments of the cells, those ions around the GPCR that's going to impact the GPCR. We know now that everything which is circulating around has an impact on the GPCR, can be an ion, can be another molecule. It would be allosteric modulation of that GPCR, and that's key to understand its biology. So if we could have the endogenous type of system to understand the GPCR and be able to express it higher in that system, that would be perfect. And we have a perfect tool to work on GPCR. Awesome. But we're not there yet.
Yeah, well, we're getting there. And for anyone listening to this, if you have a good tool,
please contact me, send me an email.
Exactly. Yeah, well, I think you raised a couple of great points about the expression and the cellular environment in a native tissue. And if we had the tools to, compare normal tissue versus disease state tissue at an expression level that allows to I think more surgically or better understand, like really map out what's going on, that would help discover more and better drugs that are more specific and more efficacious.
Yeah, indeed. And really understand, how to tackle this GPCR in that system. Yeah.
So let's switch gears a little bit and talk about junior scientists. And if you had advice to share with young scientists aspiring to contribute to the GPCR field, but also let's take it a little bit broader to improve human health. Do something that would benefit humans. What would that be? I know it's a tough one. It's a tough one.
The first thing I would say to junior scientists is that follow your heart. We choose science, like I said, not because it's going to make you rich or because it's easy path. It's a difficult path most of the time because science is not easy. So you make it because it's a passion. You really love the science. So follow your heart, what you want to do. And once you follow your heart, don't, try to enter a box. Think outside. Think, you're a scientist, your brain is working. So with all the tools there is, never limit yourself. It's today obviously difficult to follow everything that's coming out, but, try to think outside the box and coming with new ideas. Even being in a company, come up with new ideas. If you boss, I would say, like my first or second CEO said, if your boss is blocking you to evolve, talk to that person. If that person is not doing anything, just leave, go somewhere else. Today there is so many biotechs. If it's not the biotechs, you feel that you can develop yourself, you can think differently and bring the ideas you want, just go to the next one, because biotechs, they're looking for people. It's not you looking for biotechs. So it's the way the young scientists should think. They should not think, okay, I've found a job, I should stay on that job. No, you don't like that job. You don't feel that you can develop yourself. You can bring your ideas. You can change things you want to change. Go to the next one because someone else will maybe like your way of thinking. Exactly. So it's really the advice I would give, that people stop thinking that companies that they after companies, it's companies that are after them. So it's the way they should think.
Agreed. And I really like the fact that, you mentioned if you don't like it, just move on to another company. It reminded me, I think this was 2014, I was at a Gordon Research Conference, Chemotactic Cytokines, and Tom Shaw, the CEO and founder of Chemocentrics, was giving a talk. And I think it came up in the discussion that actually after his PhD, he realized that, he didn't want to work for anyone. And so he founded his own company.
Good.
Which is, it may be something that people don't think about.
Yeah, indeed.
So, step out and look at your life and see what works for you and do it. Try it.
Yeah, I think today, I think the world have so, much to offer. It's true. Sometimes the society, you can think, okay, it's hard and it's not always the best we do for the environment or for this type of thing. So if you want to change things, think of, the opportunity you have. Don't look for people to get, go and get the company you want, because you'll find what you want.
Yeah. Yeah. And I think there is such an interesting thought is, that you just mentioned, that it's just do it, just try out something. Worst case that can happen, it's not going to work out, but at least you leave with an experience. I think going out there, trying out new things, independently what the result is, will add to you as a person. You will be a richer person because you went through that experience.
Mm-hmm. Yeah, indeed.
Which I think it's quite daunting, and it's difficult. It's a difficult thing.
I think so. I think young people, what I see now, I'm coming from the previous generation, but at my time, when I was raised by my parents, is that once you find a job, you need to try to keep it. It was a way of seeing, but
yes,
I see it now. The young people think differently, and I'm actually really happy to see that there's a really want to take the opportunity, wants to impact, and if they see, oh, I cannot impact, I need to go somewhere else. I'm really so happy, and even if it happens with me, if they don't like me, fair enough, it can happen. And they decide to go somewhere to be happy. I find it nice that they have the courage to move on now. That they don't, in that mindset that we had in the past. That you have found a company, don't leave it. It's your job.
Exactly.
So it's too risky to leave it. No, now we need to be, I think in a world that we can change, and the world is changing every day anyway. So.
Yeah. And there's, I think, more opportunities. And as you mentioned, back in the day, you started a job at 20 and then you retired from the same job 40 years later.
Exactly.
Which is not the case anymore. And it's good that it's not the case, because it keeps you on your toes. It keeps you, it gives you the opportunity to find something that you really enjoy. But at the same time, it keeps you on the edge of things. It may be stressful, but it's important to evolve as a person.
Indeed. I think you today, by changing, you have really an opportunity to evolve. And as a scientist, we should never forget that it should be evolution all our life. We should never actually come back in a routine type of job, because we have a brain that is, need to be creative all the time, and we should use it.
Yes. Agreed. So top 3 aha moments in your career that shaped your trajectory. It can be scientific, it can be something else. I think one of those, while you think about it, was the fact that you felt too, quote unquote, too old to do a postdoc, and then due to industry.
Yeah, it must be the first one. And then I moved to industry. The second one, it was to leave domain and to go to Galapagos, because I have to say that many of my career so far and success was at Galapagos. Mm-hmm. And obviously, I would say it's the day where my molecule arrived to the market. So that's what. So I think this, when you realize that you made something that sick people are taking, and I think when you arrived to the market, I did not fully realize, but one day already at Confo, so the molecule I made was at Galapagos, but one day I was at Confo and I was calling okay, well, in inflammation, and we are discussing, and at some point I say, yeah, I was involved in the filgotinib discovery. He said, oh, that's a great molecule. I give it to my patient and they feel so good. And I was like, wow, it's, you just realize suddenly that, oh gosh, I did something and people are, getting better out of that thing. And I think that was the best moment, just discussing with that clinician that was telling me that, yeah, he used my molecule and people liked it and felt better with it. And I was like, oh yeah, that's, it's, you reach one of the aim of why you've been doing it. So that was one of the best thing. And I would say obviously now Confo reaching phase 1, it's one of these moments.
Of course, yeah, because it's a culmination of all that time, work, effort, money, you name it, into, and at some point it's exponential. Measuring the impact of your work by putting that molecule out that you just mentioned, it's exponential. You just, you won't have a list of people who actually are better by taking it. You can only think about the fact that you can't count them on one hand or on two hands.
Yeah,
which is very rewarding.
Yeah, it is. Yeah.
Awesome. Last but not least, you mentioned, having PhD trainees at Confo and the company growing. If and when you have job openings, where can people find these?
Most of the time, actually, it's on our website, and there is also a Flanders bio, so it's in Belgium. And so if we want, key position, like imagine protein scientists that we know that they're not specifically from Belgium, then we will try to target some, specific site. First, LinkedIn, that will be one. And we will use Headhunter. We can use a site like, I don't know if you advertise job, but I would be happy to share, position at Confo, or it's something until now we haven't done, but it's, we had difficulties to build a strong protein science team, because obviously Belgium is not known for its protein science, specifically on GPCR. So we had to bring them from all over the world. And that was been a tough time. And it was, 3-4 years ago. Now we have really world experts. But indeed, I was maybe missing a bit of a site where to post the job advert and to find these people.
Okay. Yeah. So we so far we've been accepting, it's a Google form that you fill out and we put it on drgpcr.com/jobs. But now that we're switching to the new ecosystem, that's going to be the next level where people will be able to. So there's 2 layers. One, the sign up, where you sign up and then you get approved because you're in the GPCR field. And then getting full access to the ecosystem will be membership based. Because at some point you have to be able to maintain all the infrastructure that goes with it, in general. And only site members will be able to, people who have a membership will be able to post and see the job ads. But if you sign up, you can see the listing. And the whole idea is really to concentrate the field in one place. So hopefully you don't even have to put up a job listing. You just have to look through the site members and be like, oh, you're actually, I looked at your profile on the ecosystem and you might be a good candidate. Let's talk.
Oh, yeah, that would be good, actually. Yeah, indeed. Yeah,
so that's definitely one of the next steps, because it's important to be able to, find the right, it takes a lot of time and energy and then money to find the right person for the job. So if we can make it a little bit easier, that's definitely a good.
Yeah, and GPCR World is requesting a GPCR expert. So.
Yes, exactly. I think we do live in a GPCR world, scientifically.
Yeah, indeed.
At least I do.
Sometimes I do a bit also.
All right. Thank you so much, Christel, for your time. I really enjoyed our conversation.
Thank you, Emina. It was really nice. Yeah.
All right. I'll talk to you soon. Thank you for joining us and listening in to this Dr. GPCR podcast episode. I'd like to thank our team members, Attila Forrest, Ines Pinero, and our newest Dr. GPCR protege, Montserrat Avila Zozoya. Welcome to the team, Monse. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at any time at hello@drgpcr.com. Until next time, stay safe.